Hello and welcome to this week's What Were They Thinking? The news items we have for you this week include everything from the effect of vaccines on pregnancy and the longevity of immunity in children, the newest insane conspiracy theory from anti-vaxxers regarding mRNA vaccines, the unexpected and bizarre fallout of the Russo-Ukrainian war, and other news items you can find at the timestamps given in the description box below. Let's first look at the benefits of being vaccinated if you are pregnant. The general position has been, if you are not vaccinated and looking to become pregnant, you should get the COVID-19 vaccines. Even before COVID, it was established that antibodies present in the mother will be passed through to the baby. This can also be done via breastfeeding. This means both mother and child are protected from certain diseases if vaccinated. It's one of the big reasons why influenza vaccines are strongly encouraged. There was for a time concern by various dubious quarters that COVID-19 vaccines might have an adverse effect. This has never been substantiated, and in fact, the exact opposite has been found. While the numbers are quite small, about 380 babies were looked into, the evidence is interesting and it is, as stated, consistent with past research at other vaccines and how the immune system operates. What they found from those 379 babies was that those who came to hospital with COVID and those who came to hospital without COVID, the vast majority of those with COVID were from parents who had not been vaccinated. The definition of not vaccinated is something to be aware of here, as it's not just no vaccine. It's those who had an mRNA-based vaccine and had only had the first 2 doses and not a booster. So yes, the vaccines are good if you are pregnant or attempting to be pregnant. Next is the truly mind-boggling and bizarre claims from anti-vaxxers that mRNA vaccines can somehow or other change DNA through reverse transcription. This would normally take a while, but thankfully we do have videos that you can look into to understand what reverse transcription is, so we'll provide the short version for now. mRNA is RNA that's intended to go to something like a ribosome and lead to the production of protein. It's basically a letter with a set of instructions in it. The way you would normally transcribe this and turn it back into DNA is that you would get matching nucleotides and the appropriate catalysts and enzymes. They would then create the DNA that matches the mRNA. That's how you create complementary DNA, which is a kind of DNA. The vaccines don't have that ability because there's no enzymes or catalysts present, nor are the nucleotides needed present for it to be able to transcribe it and turn it back into DNA. The mRNA, however, can be read by things like ribosomes and turned into protein. This allows for the production of things like the spike protein found on viruses, thereby letting the immune system begin to recognize that there's something foreign that it needs to be able to deal with at some point. Because this is mRNA, it has a finite lifetime, and in a manner of speaking, it's like trying to continuously photocopy something. So long as you are using a photocopy that will wear out when exposed to light, you'll find that the quality deteriorates, and at a certain set point, RNA is so deteriorated that it triggers a response from the cell to break it up and recycle the parts within it. Knowing this, we can now talk about the article in question that triggered some of this crazy thinking that somehow or other vaccines modify DNA. To start with, it's an in vitro study, meaning it's cells, not a living organism, cells. And the people who developed the vaccines went from cells to animals to humans and haven't found this, so that means their research is already starting off on a very bad foot. mRNA is present in all living tissues and cells. It's a necessary part of the genomic material, your DNA, being turned into instructions for the cells and the rest of you, or the cell itself, being kept alive. You should not be surprised to find mRNA in the cell. Further to that, the authors don't give any reason by which the mRNA vaccine would get into the cell and be reverse transcribed to become cDNA and then DNA in turn. If this were true in any way, shape or form, the food you eat could, for instance, turn you into beef if you ate hamburgers, or a pig if you ate bacon. They're just simple examples, let alone if we talk about large amounts of rice, potatoes, pasta. And whilst yes, some people are effectively a potato, such as the authors of this article, most people are not, despite probably consuming their own body weight in potatoes, if not many times their own body weight, a potato themselves, as should happen if this was taken to its logical extreme. Of course, all of that has just assumed that somewhere along the way there was, in fact, evidence to support the conclusion that somehow the vaccine transcribed itself and became DNA. Even that wasn't found. There was a small amount of DNA amplicons found, which, again, not unusual, and there's no mechanism to explain how these may have played a role in transcribing mRNA. The final nail in the coffin for this study, which is probably the most scathing flaw in it, they used a particular cancer cell line, otherwise known as immortalized cells. These cells will keep dividing indefinitely, so long as you provide them with suitable media and remove waste material. These cells also have a much higher production of RNA for both metabolism reasons and the production of proteins. You remember that thing we mentioned earlier, how mRNA plays a role in making protein? Yeah, metabolism also plays a role there. So, RNA regulates metabolism to then help to control protein production. They found a high amount of mRNA in cells that should have a high amount of mRNA. This is nothing to do with vaccines. It's simply somebody who somehow has forgotten all of their basic biology. Still on COVID-19 news, but going from the stupidity of some people to the bizarre and brilliant investigations of others. This time, how it is that COVID-19 seems to mutate the way it does, leading to the new variants, some of which have been of concern. The work comes out of the Doherty Institute in Australia. What they found, in short, is that in a very short period before the virus emerges as a new variant, there is a huge increase in the background number of mutations. For this virus, it's generally thought that there should be about 2 a month. During this period of rapid increase, that can be up to a 4-fold increase, or 8 very specific genes that can change. These genes can then give rise to the newest variants that we're now having to deal with. They did note that there has been a slow but very clear increase between each variant and how little time is taken for it to emerge. For instance, the first took about 17 weeks, while Alpha took 14 weeks, and then the next after that seemed to take a few less. The Beta variant only took 4, although admittedly Delta did buck this trend, taking 6 weeks. Nonetheless, it's unusual that rather than being a constant amount of background mutation leading to new variants, COVID-19 has exhibited episodic but very substantial periods of mutation, and the reason isn't exactly known. It is suspected that there may be natural selection at play. That is, the pressure for more mutations, therefore a greater spread and reinfection of people, could drive new variants because they can infect the hosts that past variants have infected, but they also have much greater spread. It could also be that, say, immunocompromised individuals could also be acting as reservoirs. They aren't able to clear the virus from their body, and the virus has an opportunity, while in them, to mutate and change, and then find new hosts. Going from diseases that you will acquire to diseases that you inherit. Specifically, sickle cell disease. This is a disease where your red blood cells become malformed because a protein isn't expressed properly. This causes them to turn into a crescent-like shape, hence the name sickle cell disease. These red blood cells aren't as efficient at carrying oxygen around the body because they tend to get stuck. When that happens, you can get blood clots. Your immune system sees them as foreign and actively tries to remove them. And, of course, being an irregularly shaped cell, it can cause all sorts of other problems, especially in the tight confines of capillaries. Because of all that, you can imagine how it's something that people are trying to treat. Last week alone we mentioned how you could have genetic therapy and theoretically resolve it. While that's still being worked on, other, more effective treatments have to be brought out. In some cases, you can be bled and have blood removed, just like a donation, but it's simply going to be discarded. Then have an infusion or transfusion of blood from a donor. This replaces a large amount of your otherwise dysfunctional blood with functional blood. Another option is to have the immune system trained to pick up on these red blood cells and remove them, but that could lead to all kinds of problems with regards to autoimmune diseases. So that doesn't work, but something very similar can: the use of monoclonal antibodies. The monoclonal antibodies are beneficial in that they flag the sickle cells as being undesired, but they make sure to only do it in such a way that the immune system doesn't go on a rampage and start destroying all red blood cells. This is one treatment that's actually been around for quite a while, roughly 45 years or thereabouts. The problem from the point of view of getting this to market has been approval. Once that hurdle was overcome, it could reduce the chances of sickle cell crisis by large margins, in some cases as much as 60%. Specifically, we're talking about a condition, or at least a subset of a condition, called sickle cell crisis. This is when the red blood cells begin to initiate that immune response and create a clotting cascade. That clotting cascade leads to them clumping together and blocking the flow of blood. By using certain monoclonal antibodies, you can block platelets, blood vessels, and a few other involved proteins from interacting. When this happens, you reduce the number and nature of the clots that are formed, thereby reducing not only the risk of a sickle cell crisis, but also the impact of the disease overall. The article that this information came from can be found in the description box below, and it provides much more information on not only the specifics of one particular treatment, but the development and how it's both a good and bad thing. Going from the medical news to technology developments and an incredibly tiny battery. Just to give some context, the battery is smaller than a grain of salt. Not a grain of rice, not a grain of sand, a grain of salt. It's so small, and yet it contains enough energy to run the chipset that it was associated with for about 10 hours. Not only is this incredibly tiny battery able to run for so long, it's rechargeable. And that distinction is important as we are moving ever closer towards tiny, most likely autonomous robots that will need to be able to run themselves for a long period of time without support. The battery is unique as opposed to others in the way it works. The initial design is very similar to other chemical batteries. A twin layer, one positive, one negatively charged, and an electrolyte fluid in between. This allows for the movement of energy across it and therefore a charge to be generated. The problem with this design is that if we're talking about a large battery, it's going to need to be rolled up, and material at this size is going to be too brittle for that to happen. Or at least it is if you use force yourself on it. If instead, as these researchers have done, you make it so that the design of it tries to roll itself up into a spiral shape, you completely mitigate that particular problem. Going from research that's groundbreaking to the breaking of research. As a result of the fallout of the Russo-Ukrainian War, Russia has just declared they are ceasing any joint experiments on the ISS. This is honestly not that much of a surprise, but nonetheless, it is undesirable. Not only will Russia now cease cooperating with the European and North American countries that are involved with them, they're also going to cease the supply of rocket engines and the maintenance of those rocket engines that have already been delivered. It seems that international politics don't stop once you leave Earth. That's all the news we have for you this week. Thank you for watching. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you might have below.